Hello everyone, this is a full walkthrough of the Pearson Edexcel International A level. This is for Unit 1, so this is considered AS level. For Molecules, Diet, Transport, and Health, this is for the 8th of October 2024. Let's get started. Okay, so question 1. Amoeba are single-celled organisms that live in water. They are able to change their shape. The diagram shows two different shapes that one amoeba can make. We have shape 1 with a diameter of 300 micrometers, and then we have another shape, which is shape 2. So we can see a cell membrane, a nucleus, and a cytoplasm. This looks very similar to an animal cell, where we have a nucleus, we have a cytoplasm, and a cell membrane. So an amoeba changes shape by pushing cytoplasm against the cell membrane, forming long, thin projections. Okay, we get that point. State the property of the cell membrane that enables the amoeba to change shape. Well, remember, this is a picture, this is a diagram of the cell membrane. We have a channel protein over here, and a channel protein is also called an integral protein because it goes through the cell membrane. So how can this amoeba change shape so easily? Well, the cell membrane is fluid. This is why sometimes it's called a fluid mosaic. The cell membrane is fluid. The cell membrane is very fluid and it can move laterally. It doesn't move up and down, it moves laterally. Okay, so gas exchange in amoeba takes place across its cell membrane. The amoeba can be roughly spherical, as shown in shape 1, which is the surface area for the amoeba in the shape. Use the formula surface area equals 4 pi r squared. Now, in the question before, the previous question, we had a diagram where you've seen the picture of the amoeba with the diameter being 300. So the diameter was like this and it was 300. Now remember, the radius is half of the diameter, so it was drawn out like this, and it's going to be 150. Okay? And also notice how in the question they put pi over here. So this means that the pi is not going to be incorporated in our calculation, because if I incorporated pi, it's going to give me 282,743. But with pi, it's only going to give us 90,000 pi, which is the same number as this one. The only difference is that we have pi over here and no pi over here. So therefore, our answer is going to be 4 and then brackets 150 to the power of 2, which gave us 90,000 pi. Therefore, it's A. This is one question that you should not get wrong. Now, question double I, the mass of the amoeba stays the same when it changes from shape 1 to shape 2. Which row of the table shows what happens to the surface area and volume of this amoeba when it changes to shape 2? They always like to bring questions about surface area and volume. This is like a repeated idea in IGCSE and in A level as well. Okay, so I like to think of it as a balloon. When you have a balloon and you squish it, okay, the volume inside the balloon is going to stay the same. Okay, so if you have 1 cup of water in a balloon and you squish it, and obviously you tie the balloon, it's still going to be 1 cup of water in the balloon. However, the surface area of the balloon is going to stretch. Okay, so the surface area is going to increase, like the balloon. When we squish it, it stretches, so it increases, and the volume will stay the same. Therefore, the answer is going to be C. Okay, explain why an amoeba can rely on diffusion to supply the oxygen it needs. Well, remember that diffusion, the oxygen is going to go from a high concentration to a low concentration. Therefore, it's a passive process, meaning it does not require any energy. So the first point should be because there is a higher concentration of oxygen outside of the cell. Okay, the second point is because amoeba have a large surface area to volume ratio. You can also mention how oxygen is nonpolar, therefore it will go through the membrane without being interrupted by any forces. However, just stick to these 2 points as they're much more simpler and much more clear. Now, question 2. During the development of a human, the structure of hemoglobin changes. In the developing fetus, the main oxygen-carrying molecule is fetal hemoglobin. Okay, remember hemoglobin is a globular protein. Okay, just remember that. Towards the end of fetal development, fetal hemoglobin is replaced by adult forms of hemoglobin. So blood may contain 5.3 million red blood cells in 1 millimeter cubed of blood. One red blood cell can contain 270 million hemoglobin molecules. Calculate the number of hemoglobin molecules in 1 millimeter cubed of blood. So express your answer in standard form. Even if they didn't ask you to express your answer in standard form, it's always better to write it in standard form. So since it's million, we're going to have 10 to the power of 6, because a million has 6 zeros, so it makes sense that it's going to be 6. So we're just going to simply multiply them together. So 5.3 times 10 to the power of 6 in brackets times 270 times 10 to the power of 6, which gave us 1.3 times 10 to the power of 15. Now that's a really huge number. This is why it's in standard form. Okay, now make a flashcard of this table. This is the table that I used when I used to do my A-levels, and this table really helped me to understand the differences between fibrous proteins and globular proteins. Okay, so which of the following statements about the structure of hemoglobin are correct? So it is a fibrous protein. No, that is wrong, because an example of fibrous protein is collagen and keratin. Now, these are things found in your skin and in your hair and in your nails, okay? Fibrous proteins are usually long and narrow, and they're mostly insoluble. They are also less sensitive to changes in heat and pH, okay? So fibrous proteins are like stronger, okay? Now, is hemoglobin a quaternary structure? Yes, hemoglobin is a quaternary structure. Why? Because hemoglobin has a range of primary, tertiary, secondary, quaternary, all of them mixed together. Okay? It contains a non-polar heme group. Yes, hemoglobin has 4 non-protein heme groups. So therefore, it's going to be 2 and 3, so the answer is D. And also make a flashcard of this table. It's very important. Okay. The graph shows the changes for 3 types of hemoglobin subunits present during each stage of development. So when a fetus is going to have a different type of hemoglobin than it will when it's like 5 weeks old or 10 weeks old, or even when it's given birth. This is because the way that the fetus takes in oxygen from the mother. At each different stage of development, the way that the oxygen is delivered to the fetus is going to be different. Okay, therefore, chemically speaking, the hemoglobin is going to have to be different. So it's going to have to either take so much oxygen at one point, or it's going to have to take lower amounts of oxygen at another point, which is usually the later stage. Okay, so we have the alpha subunit, the beta subunit, and the Y subunit. Alpha is A, beta is B, and I hope you understand why. So, describe the changes in these types of subunits present during each stage of development. So I color-coded this so that you can see very well. So we have the green, they all increase. So during the embryonic development, alpha, beta, and Y subunits increase. Now the second one is the B, where they're more constant. So in fetal development, alpha is constant, beta is constant, and then increases, whereas Y decreases. You can see Y's decrease over here, and beta's slight increase over here. Okay, now during the baby stage, alpha remains constant and beta increases, then levels off. Y decreases, then remains constant. So when they give you a question about any graph or any data, make sure to mention the slightest point. So you might say, oh, the majority over here is constant, but this part there's a slight increase, so I'm not going to include the slight increase because it's very small. No, always include everything you see. If it's constant for a bit and then increases for a bit, include that. If it's increases and then remains constant for a bit, also include that. Okay. Okay, the oxygen affinity of fetal hemoglobin is different from adult hemoglobin. Explain why this difference ensures the fetus obtains enough oxygen during fetal development? This question was also repeated in my A level, so I would just like you to make a flashcard of this. So just pause the video, make a flashcard so that you can use it while you're studying. Okay, so number one, fetal hemoglobin has higher affinity than adult. Why does fetal hemoglobin have higher affinity? So that it can take more oxygen, okay? Because it requires oxygen for development, okay? Number two, so that oxygen will leave the adult hemoglobin and bind to the fetal hemoglobin, okay? So the hemoglobin wants to bind with as much oxygen as possible. Okay, question number three. CMT is an inherited disease that results in the gradual loss of function and sensation in hands, legs, and feet. Over 100 different gene mutations have identified that cause CMT. The pedigree diagram shows the inheritance of one form of CMT in part of a family. The allele for CMT can be represented by the letter C in this recessive pattern of inheritance. So that means that if you have CC lowercase, which is recessive, you're going to have CMT. Okay, so since child 4 has CC recessive, this means that parent 1 and parent 2, they both must be characters. So they're going to be heterozygous. Heterozygous meaning different. So capital C, lowercase c. Okay, both parent 1 and parent 2 are heterozygous. Okay, they have one dominant C and one lowercase c. Okay, which row of the table shows the genotype and phenotype for individual 1? Well, individual 1 is going to be, the genotype is going to be capital C, lowercase c, since it's a carrier, okay? That if it's not a carrier, if it has double C, and so if it is homozygous dominant, the question wouldn't make sense, okay? Or else child number 4 would not be affected. So number A is not possible. Number B, however, is possible because it is not showing CMT, however, it is a carrier, okay? Therefore, the phenotype is unaffected heterozygous, so it's B, okay? Which allele combination could individual 3 have? Well, just draw out a Punnett square. We know parent 1, parent 2 are both carriers. Draw out a Punnett square. Therefore, it's going to either be capital C, or C, lowercase c. Therefore, it is D. Okay? What is the probability that a 4th child would be a female with CMT? I used to be so confused with these questions because I was like, do you add them? Do you multiply? No. You just multiply them, okay? So the chance of being a female is obviously 50%, okay? You're either going to be a male or you're either going to be a female at birth, okay? So therefore, the chances is going to be 1 over 2. So we're just going to write female 1 over 2 and then times, okay, the chance of getting CMT, which is going to be 1 over 4 as we found out from our Punnett square. So 1 over 2 times 1 over 4 is going to give us 1 over 8, because 1 times 1 is 1, 2 times 4 is 8, 1 over 8, which gives us 1 and 8, or it can give you 0.125, which when you click on your calculator, there's a specific button that you click on. It starts with S, I believe. It will give it to you in a fraction. So it's going to be 1 in 8. Okay, other forms of CMT show a dominant pattern of inheritance. In the USA, 5% of people with CMT have one of these dominant forms of the disease. So in the USA, there are a total of 126,000 people with all forms of CMT. Calculate the number of people in the USA who have one of these dominant forms of CMT. So we're just going to want to find 5% of 126,000, which gives us 6,300. You can also write that in standard form, which is going to be 6.3 times 10 to the power of 3, because we have 1, 2, 3. And if we have a decimal point over here, that's 1, 2, 3. So 3 over here. Okay. An individual with a dominant form of CMT will have the allele combination T. Remember, that's going to be homozygous, dominant, because they're the same, homo, same, okay, or capital T, lowercase t, which is heterozygous, hetero meaning different. So, draw a genetic diagram to show the possible allele combinations of the children of the following parents. So one parent does not have it, which is going to be lowercase t, lowercase t. Parent 2 has the allele combination capital T, lowercase t. So we have our genotypes over here, and then we draw our Punnett square, which is going to give us the possible phenotypes. So it's a 50% chance of having it. So because the capital over here is going to be affected. So it's 50% chance affected, 50% chance unaffected. Okay, so another form of CMT can be inherited as a sex-linked trait on the X chromosome. Okay, explain why a male CMT cannot pass the disease on to his sons. Okay, so a male has XY chromosomes and the CMT is only carried on the X, okay? His sons are going to be XY, meaning that the male is only going to pass down the Y chromosome and the female has no option but to pass down the X chromosome, but since it's a female, it's not going to have the CMT disease on it because it's only on the male, okay? So the CMT allele is only on the X chromosome and only the Y chromosome is passed on to their son. Okay, so question number 4 is about the circulatory system. So, the circulatory system and the cardiac cycle ensure that all the cells of the body are supplied with their requirements. The table shows the diameter of the lumen of an aorta and a capillary and the thickness of their walls. So we have the type of blood vessel in the table, which is the aorta and the capillary. The diameter of the lumen of the aorta is 2.5 centimeters, which is fairly larger than the capillary because remember capillaries are very small. And then the thickness of the wall is 2 millimeters, whereas the thickness of the capillary is 0.001 millimeters. Okay, how many times greater is the diameter of the lumen of the aorta than the diameter of the lumen of the capillary? So the first thing that we have to do is we're going to have to ensure the same units for our calculation. So from centimeters to micrometers, you're going to have to multiply by 10,000. Now we're going to have 2.5 times 10,000 divided by 0.14 micrometers for the capillary, which gives us 179,000. Therefore, the answer is D. Now, this question is a repeated question in GCSE and A-level, so make a flashcard of it. So, explain why the wall of the aorta is thicker than the wall of the capillary. Well, the wall of the aorta has elastic fibers because the blood is under high pressure from the heart. Remember that the aorta, so A equals away, because the aorta is going to pump blood away from the heart. This blood is oxygenated and the aorta has to have very thick muscles and walls to push out blood in the highest amount of pressure possible to ensure that oxygenated blood can reach the heart and all the way down to your feet. The second point is, or the third point actually, the capillary wall is only one cell thick so that efficient gas exchange can occur. Okay, the table shows the durations of a ventricular diastole and a ventricular systole at two different heart rates. Remember, diastole is closed, systole is open. Systole, diastole, diastole, systole, okay? This is just the opening and shutting of the valves. So the heart rate is beats per minute. We have 75 and 200. We have the duration of ventricle diastole per second and duration of ventricle systole. So here we're talking about the duration of the valves being closed or the duration of the valves being open, not the rate of how fast they open and close. No, the rate they stay closed or the rate they stay open. So determine the relationship between the heart rate and the duration of ventricular diastole and ventricular systole. Well, the ventricular diastole and systole decrease with increased heart rate. Why does that make sense? Because we are talking about the duration. So duration of the valves being closed or the duration of the valves being open. They're going to open and close very fast. Therefore, the duration of them opening very fast is going to decrease with an increase in heart rate. Okay, ventricular diastole decreases by 0.39 seconds. It's very important that when they give you a graph, to ensure to get the second point, manipulate the values in the graph and use them to your advantage to further explain and demonstrate your knowledge. And you will also get a grade from it. The graph shows the rate of flow of blood through the aorta of a dog with increasing distance from the heart. Okay, so we have the rate of flow of blood in the forward direction, and we have the rate of flow of blood in the backward direction, and we have increasing distance from the heart. As the distance from the heart increases, obviously the rate of flow of blood is going to decrease, and notice how over here we have some backflow. So explain the changes in rate and direction of blood flow through this aorta. Use the information in the graph to support your answer. Well, the peak rate of flow in forward direction decreases with distance from the heart. The rate of flow decreases due to lower pressure. As the blood goes farther away from the heart, there's going to be less pressure. I hope that makes sense and why. Okay, there's some backflow of blood which corresponds with diastole. The further the distance away from the heart, the lower the pressure as the aorta is branching. Okay, so now it's question 5. Warfarin is used as an anticoagulant to treat some patients. So remember, you're going to need to memorize some different drugs. So anticoagulants, they just prevent the blood clotting process. So someone with high blood pressure or someone who smokes a lot is going to be taking a lot of anticoagulants to prevent any blocks in the coronary arteries. So patients respond differently to this drug. This makes it difficult for doctors to select a safe but effective dose of warfarin to give to each patient. Factors that affect the response of a patient to warfarin include age, body mass, liver function, and genetic makeup. Explain why a safe but effective dose of warfarin needs to be given to a patient. Okay, so not too high a dose, because to avoid uncontrollable bleeding, and at the same time not too low a dose to avoid blood clotting. There are two genes that code for proteins that affect warfarin and the blood clotting process. We have gene M, which codes for enzyme M that breaks down warfarin into inactive molecules, has many alleles, and two of these codes for much slower breakdown of warfarin. We also have gene V, which codes for enzyme V that converts vitamin K into an active form that triggers the blood clotting process, and one version of this gene results in a decreased production of the messenger RNA, which is mRNA, needed for the synthesis of enzyme V. Remember that enzymes are proteins, so they will require mRNA. Okay, describe the role of mRNA in the production of enzymes. They could also ask you, describe the role of mRNA in the production of proteins. These two questions are perfectly fine, and you must know the function of mRNA and its role. So, mRNA is needed to carry a copy of a gene out of the nucleus, where transcription occurs, okay, and then used for ordering of the amino acids. Okay, Pharmacogenomics analyzes the genetic profile of individuals. This analysis is used to determine the appropriate doses of a drug for individual patients. Discuss the possible use of pharmacogenomics in selecting appropriate doses of warfarin for individual patients. Use the information in the question and your own knowledge, hint on your own knowledge, okay, of the blood clotting process and genetic screening, and to support your answer. Okay, so the second they said of the blood clotting process, I'm gonna include a sentence down somewhere where I'm just explaining what the blood clotting process, because I'm 100% sure that this is gonna give me at least 1 mark. Okay, so pharmacogenomics may identify the genotype which determines the dose of warfarin needed, meaning different doses of warfarin will be needed for different patients, since two genes from two separate people have several different alleles. Okay, pharmacogenomics may identify high cholesterol or high blood pressure that is passed down by genetics, because remember that high cholesterol and high blood pressure can also be caused by genetics. However, genetic screening may highlight ethical or religious issues, or even families may not want to find out about any underlying genetic issues. Okay, so warfarin prevents blood clotting where thromboplastin converts prothrombin into thrombin, which converts fibrinogen into fibrin, which forms a mesh thing to trap platelets. So notice how I included blood clotting process down here, and I've included ethical issues, and I've included possible uses of pharmacogenomics. Okay, question number 6. The photograph shows some soybeans. We have soybeans over here, just a general picture. So, eating soybeans may reduce cholesterol and low-density lipoproteins, LDLs, in the blood. Explain why eating soybeans may reduce the risk of heart disease. Well, less cholesterol, so this will reduce the formation of atheroma in the coronary artery. Okay, it's important to note that atheroma occurs in the coronary artery, okay, meaning blood flow to the heart tissue will not be stopped. So this means that oxygen is still available for aerobic respiration. Okay, the extent to which soybeans reduce cholesterol depends on the variety of soybeans. This reduction is influenced by two proteins found in soybeans, glycinin and beta-conglycinin. Give two ways that the primary structure of two proteins could differ from each other. Okay, so remember that our protein is basically like a huge chain of amino acids. So remember, they can differ that the total number of amino acids is different, or the sequence of amino acids is different. So they can have 5 amino acids, or they can have, for example, lysine, glycine, ricin, and then they can be in a separate order. Okay, so that's what sequence means. Okay, so the effect of soybean extracts on cholesterol production by liver cells growing in culture was investigated. Scientists produced fat-free soybean flour and digested it with enzymes in a way that copied digestion in human digestive system. The diagram shows some of the steps used. We have step 1, where's the mouth. Soybean flour is mixed with saliva to form a moist ball of flour. We have step 2, step 3, step 4, and step 5. Okay, so explain why the scientists used digested soybeans to make the extract for this investigation. Well, they want to mimic what is happening in the body. Okay, and it's also needed to extract the proteins in soybeans. Remember, our saliva has amylase, which breaks down the extracts in soybeans, which extracts out glucose and proteins. Okay. Question II. Suggest why the soybean flour was incubated with different enzymes in step 2 and 3. Well, remember, our digestive system has different enzymes in every part. The enzymes in our stomach are not the same as the enzymes in our colon, or our enzymes in our large intestine or our small intestine or in the duodenum. Okay, so because different enzymes are found in different parts of the digestive system. Okay, now, question III. Explain why the soybean flour and enzymes were incubated at 37 degrees for 2 hours in step 2 and step 3. So 37 because this is the optimum temperature for human enzymes. Remember, if someone has a fever, their body temperature is going to either be way above 37 degrees, meaning they are sick, okay? Now, why is it for 2 hours? Well, this ensures enough time for complete digestion to occur. Okay, the soybean peptides at different concentrations were incubated with the liver cells growing in culture. The enzyme HMGCR is involved in the synthesis of cholesterol. The effect of soybean peptides on the activity of this enzyme and the synthesis of cholesterol was measured. The graph shows the results. We have our percentage on our y-axis and the concentration of soybeans of peptides on our x-axis. Now notice how as the inhibition of HMGC activity increases, our synthesis of cholesterol decreases, therefore that there is a correlation between what's happening. So explain why eating soybeans may reduce cholesterol level in the blood. Use the information in the graph to support your answer. Well, as the concentration of soybean peptides increases, the inhibition of HMGCR increases and the synthesis of cholesterol decreases because soybean peptides inhibit HMGCR. Since HMGCR is inhibited, the cholesterol precursor cannot be converted into cholesterol, so less cholesterol is being made, which is better for your health. Now, question number 7. Scientists investigated the numbers and types of mutations in different species of animals. Okay, samples of tissues were collected from living animals of each species and the type of mutation determined. The types of mutations were classified as either substitution mutations or insertion and deletion mutations. The table shows the results for this investigation. We have a naked mole rat, we have a mouse, a dog, and a human. Okay, so describe how these types of mutations can affect DNA-based sequence of a gene. Well, substitution mutations, it's in the name. Okay, substitution is the same thing as swap. So substitution mutation, they swap nucleotides. Okay, and insertion and deletion mutations result in a change in the number of bases. This is really important for you to know because this is going to be asked in your exam. So make a flashcard of this question as well. Okay, this question is also really interesting and is repeated in most of the exams I have done. So, explain why these animals can survive with a greater number of substitution mutations than insertion and deletion mutations. Okay, so look at this over here where I'm circling. You see how we have UCU, UCC, UCA, UCG. All of them code for the SER amino acid. So if we substitute UCU, if we substitute the last U for another C, it's still technically SER amino acid. Okay, I believe it's called serine. So it's still the same amino acid. Okay, therefore technically there's nothing changing about it. Okay, so substitution mutations will not affect protein function, as substitution mutations may not alter the amino acid sequence. Okay, they like the word sequence, so make sure to include it. Also, the substitution mutation may occur in a non-coding region. And if you see a real amino acid sequence, it can be very long. However, many parts of it are non-coding and they don't do anything. Okay, so and also deletion mutations may have a large effect on protein function as it causes a frame shift. It changes the function of the amino acids and it brings in different amino acids, therefore changing what's actually happening. Okay, suggest which set of data is most valid. Well, it's going to be the mouse, since with mice, you can sample a larger amount. Therefore, it's more valid. Okay. So, the graph shows the number of mutations per cell during the lifespan of these 4 animals. Calculate the mean mutation rate for the mouse. Draw a line of best fit onto the graph to help you. Well, over here I drew my line. I tried to draw it between all the different points, but a different range. Usually in these types of questions, they're going to give you in the mark scheme a range of numbers that are going to be allowed. Okay, just draw a nice neat line using your pencil. So draw a best fit line to help you, and then the mean is just going to go to year 1. So over here, just draw a straight line. It's roughly around 900. I think it's gonna allow any number from here. Sorry, it's gonna allow any number from here to here. So I just chose 900 and it's correct. Now, describe 3 conclusions that can be made from these graphs. Well, number 1, the number of mutations increases with age in all 4 animals. You can see over here, there's an increase in all of them. Okay. Number 2, different species have different lifespans. That's also true. Okay. Look at age here, 16, 8, 2, 80. They're all different. So the third point, different species have different number of mutations. That's also true. Look at the amount of mutations here, and then look at the amount of mutations here. Okay. Now, triple I. Suggest 2 reasons for the difference in the number of mutations per cell of these species of animals. Well, number 1, cells divide at different rates. Number 2, cells are exposed to different environmental factors. Different environmental factors can lead to different mutations, okay? Okay, now question 8. The photograph shows 2 avocados. Avocados are high in unsaturated fats and low in saturated fats. Compare and contrast the structure of an unsaturated fatty acid and the structure of a saturated fatty acid. This is a constant repeated question in all the exams, so also make a flashcard of this. Number 1, both contain carbon and hydrogen. Okay, the second similarity is both contain covalent bonds. However, a difference is unsaturated have carbon-carbon double bonds, whereas saturated have single carbon-carbon bonds. Remember, saturated single, unsaturated double. The effects of eating avocados each week on the risk of coronary heart disease was investigated. A group of men and women who had no signs of cardiovascular disease or stroke were selected. They were all between the ages of 30 and 75. The group ate avocados each week to replace animal products in their diet whenever possible. The group all completed a questionnaire about the food they ate at the start of the investigation and every 4 years for over 30 years. That's a good period for an exam. There were 41,701 men and 68,786 women in the group. What is the ratio of men to women? All you have to do is just divide men by women, which gives you 0.6. So therefore, our ratio is 0.6 to 1. Because we have 0.6 men to women, so put the men first and then the women. Therefore, this is going to be a ratio of men. Because if you choose B is indicating that 1 for men, which is wrong because the question says men to women. So describe the questions that need to be in the questionnaire so that appropriate information can be gathered for this investigation. This is a really good 6-mark question. I think that this is one of the easier questions that they could have asked. So when we're talking about coronary heart disease and LDLs and cholesterol and food and diet, we need to talk about number 1, animal products. Number 2, how much food they eat. We can include in antioxidants. We can also include questions from the investigation itself. So they mentioned avocados. Well, how many avocados did they eat? What type of avocados did they eat? We can also mention other factors such as smoking. Okay, so the first question we can ask is, how much animal products do they consume in a week? Because animal products such as a fatty steak contain high levels of cholesterol, which increases the risk of atheroma. Notice how I gave the question, and then I explained it, and then I gave a reason. The other question can be, how much food such as kiwis and oranges do they eat? Okay, as they have high levels of antioxidants, and remember, antioxidants reduce the damage to endothelial cells. Another question is, how many avocados do they eat per week? To see if the number of avocados affects the results. And also, do they smoke? Because this wasn't given to us in the beginning of the question, because smoking increases blood pressure, which damages the endothelial lining. Now the final question, comment on the validity of the methods used in this investigation. For these questions, you can usually go that the question is valid or it's not valid. However, for the purpose of this question, I chose that it's valid. You can also say that it's invalid for multiple other reasons, which is also appropriate and also reasonable. So it's valid because there's a large sample size, okay, and it represents both men and women. And it's also ran for a long period of time. 30 years is a good amount. And also, participants in the beginning had no signs of coronary heart disease. Thank you so much for listening, and I hope this helped.